Welcome to Houston House at South by Southwest. Scientists are on the cusp of medical breakthroughs that are creating a new paradigm for healthcare. Breakthroughs that have the potential to cure even the most difficult diseases by focusing on the building blocks of our bodies, our cells. MD Anderson, one of the largest and most respected cancer centers in the world, has a long-standing history of transformative innovation in healthcare. Today, their outstanding clinical care, strong clinical trial infrastructure, and fundamental commitment to translational research continues to pioneer treatments for patients from across the globe, all while fostering a culture of innovative thinking. Join industry leaders from MD Anderson Cancer Center as they discuss the shifts and challenges facing the biotech industry, as well as the unique resources that the Houston region offers startups in this space.
Welcome to everybody to the Houston House. My name is Larry Hope. I'm the Director of Business Development for the Strategic Industry Ventures Group at MD Anderson Cancer Center. I have the both fortune and misfortune of working with these two gentlemen relatively frequently at work. And anyway, we're excited about sitting on this panel today to kind of talk about some of the innovations in both the Houston ecosystem as well as sort of the med tech and certainly cancer oncology therapies, but just the evolutions of just the innovation of those fields. Just real quickly before we get into, they have a couple of presentations, and then we're going to have a little bit of Q&A or kind of a panel discussion, and then we'll open up to the floor for anybody with questions or anything that comes up from the audience. Just real quick show of hands, just to kind of get a feel for who's in the audience. I'm just curious, are there investors in the room? Okay. Okay, thank you. People working in the biotech, biomedical space? Companies, okay. How about healthcare, maybe on the hospital or clinical side of things? A few of those. Students? Okay, we're all students all the time. Okay, no, that helps. So just to kind of get a feel, I realize these things will have a broad draw of different folks. So anyway, let's go on and move into our two speakers today. The first is Mike Curran, and I'll let him do introductions, and I think he's got a short slideshow to walk us through.
Great. Nice to meet you all. I'm Mike Curran, as Larry so nicely introduced. I am also at MD Anderson as associate professor of immunology, and I started a spin out with some technology we licensed from MD Anderson called ImmunoGenesis, which is in the field of immunotherapy, which I'm going to tell you a little bit about now. So, fire up the slides. I'll tell you a little bit about what's driving a really innovative environment in Houston, where we've seen, really a new era of startups in oncology research, but particularly focused around the field of immunotherapy. So why is immunotherapy interesting? Well, not that long ago, 2010, so a little more than a decade ago, if you had cancer, we treated you with one of these sort of three pillars or some combination thereof. There's always been chemotherapy, radiotherapy for about half of patients, targeted therapy wasn't available to very many. What these were chemotherapy. We try to knock the cancer down with poisons that prevent it from dividing. For patients with metastatic solid tumors, this would, by time, it was almost never curative. Radiotherapy, very good for removing a targeted instance of cancer, but never really useful for systemic disease. And then targeted therapy was great for some patients, let's say with lung cancer with an EGFR mutation. We give you an EGFR inhibitor. A large percentage of patients would respond. Problem is, cancer would always come back. It would always find a way to evade. So this is where we were, right? And where we are now is we've had this other pillar show up, which is immunotherapy. And the reason this has been so transformative is, still we can't treat everybody this way, but about 40% of patients can get immunotherapy. But 10 to 80% ORR means, look, if you have Hodgkin's lymphoma, I give you immunotherapy. There's an 80% chance this is really going to knock your cancer down, maybe cure you. Bladder cancer, some other things that we have harder time treating, it's 10 to 20%. But across this spectrum, we're able to treat patients, and the real change from these three other pillars is that some of the patients that respond are durably cured of their cancer forever. So for the first time, really, immunotherapy allows us to offer patients with metastatic solid tumors, the opportunity for a cure. And if where we're at right now is patients who have cancers that are hard to treat this way or or immunotherapy isn't available are looking at the ones like melanoma where this is really transformed therapy and saying, I really want that. This looks a lot better than the stuff we used to have. And so there's a huge innovation going on where companies are trying to develop immunotherapy so that we could treat more than 40%, more like 100% of patients. And just to give you an idea, within the Texas Medical Center in Houston, we have over 100 clinical trials of new immunotherapies going right now. And this is not MD Anderson, Methodist, Baylor. You've got this huge nucleus really invested in immunotherapy, and that's what's driving, at the academic level, this innovation, which is then being fueled and also, in its own way, fueling an innovative environment in startup companies. So just so we're all on the same page, very diverse group, what does your immune system do? Well, your immune system knows healthy you. It knows what yourself should look like. And when something shows up that's not supposed to be there, let's say flu. Flu is a virus people used to get before COVID. So flu, I got to update my slides now. It's historical now. But so flu has 8 small genes, just 8, right? And that's plenty for your body to see it as something completely foreign and eradicate it. How does that work? Well, you make antibodies that keep the virus from getting into any new cells. And then, and this is important for immunotherapy, the cells that have already been compromised, they've already got some flu in them, we don't want it to spread, your T cells go and they kill those and wipe them out before they can spread the disease. So it's T cells that we like for immunotherapy because they're the ones that can kill cancer cells. So healthy self, that's what your immune system does. Well, tumors have hundreds, sometimes thousands of changes, not just those 8 that we found in flu. So your immune system should be totally capable of seeing cancer and wiping it out. Huge red flags everywhere. So how can cancer even exist in the light of this revelation that it should be wildly visible to your immune system? Well, the problem isn't that your immune system can't recognize cancer. It recognizes it really well. The problem is that tumors cheat and they actively disable your immune system locally. And the way they do that, and if you think about it, cancers can draw upon this whole library of genes that exist in all your cells. If you get an infection in your eye, you're going to clear it, but you're not going to let cells in that would make you go blind, right? That's good. If you're carrying a child that is half genetically different from you, your immune system doesn't rush in and say, oh, that's different, we got to get rid of that. That would be bad for us as a species. And so there are genes that mediate these protections that say, no immune system here, not allowed. Tumors draw on exactly those genes, placental immune barrier, barrier in the eye, and they create their own local state of immune privilege. And so what immunotherapy is realizing that tumors have to do this. They have to find a way to evade or disable your immune system locally. Immunotherapy seeks to reverse those adaptations, right? We're going to restore the capacity of your immune system, which exists naturally to recognize and reject this cancer. And it's become a big tent, right? The field has grown, people see this is working. T-cell checkpoint modulation is a fancy term for there are a bunch of off switches on T cells that tumors will throw to shut off your immune system. The first drugs we have that are approved for everybody block those off switches. They deny the tumor this easy opportunity to shut your immune system off. I'm going to talk a lot about that. I'm going to leave the others, particularly this giving T cells directly on the right side, to some of my esteemed colleagues here. But why are we interested in T-cell checkpoint modulation? Well, this is, even 90-year-old Jimmy Carter could be cured of his lung cancer this way, right? So this really is something that has been widely applied. It works for a lot of people, and there's a really strong Texas connection here too that I'm going to mention. But first, I'll mention really briefly how this works. I'm not going to go heavy on the science, but it's really a logical concept in that cancers, like this tumor here, they have these genetic changes, right? And you've got these cells called dendritic cells. They'll pick up those mutant proteins, activate a whole bunch of your T cells to go and kill anything that has those mutant altered proteins. All of that is great. Everything's going like it should. What happens is those T cells, when they get turned on, though, they get these safety switches turned on, CTLA4, PD-1. And the reason those exist is, if I'm sending these T cells to get rid of those flu-infected cells when I have flu in the lungs, when the flu's gone, I want those cells to shut off, right? You have lots of killer cells that can kill any cell in your body very easily. When the threat is gone, you want them to get turned off. And they're natural control switches that will turn these off when the threat is gone. The problem is tumors are cheaters, and they surround themselves with cells that have all the triggers to turn these off immediately when they get near the tumor. And tumor cells themselves carry kind of the triggers that hit this off switch. So you send your T cells in just like you want them to wipe out this tumor, and they just get switched off before they can really do anything. So what do? We give drugs, which are really just antibodies that block those off switches. And when this happens, even as someone in the field, it can be really amazing. You have a melanoma patient comes in. I reference melanoma because he's worked really well in melanoma. You have a melanoma patient that comes in, they have hundreds of cutaneous nodules, tumor in the lungs, tumor in the liver. 12 or 16 weeks later, blocking these off switches has allowed those T cells to get in, wipe out those cancers, and there's no cancer left. They're gone. So the power of your immune system when fully unleashed, even against a bulky metastatic cancer, is there. It's strong. It can wipe it out. But you really need this full sort of licensing of your immune system to go all out. Now, why doesn't this work for everybody? Well, it turns out we have problems. Some tumors are really good at keeping T cells on the outside. They basically build a wall. We can't get them in. No matter how much we activate them, they can't do the job. And also, tumors are good at sort of poisoning their environment, so T cells can't live there very well. These are problems we're dealing with now. But I wanted to go back historically. So I had the pleasure of training with Dr. Jim Allison, who is a Texas native, also works at MD Anderson right now, won the Nobel Prize for discovering really the underpinnings of most of the immunotherapy we do now, which is immune checkpoint blockade. And this, back in 1996 was the real revelation. And keep in mind, at the time, all we knew then was really chemotherapy and radiotherapy for cancer. He showed that blocking just one of these immune checkpoints, just one of these ways that cancers could switch off T cells was enough. Tumors grew, then they went away. And when they went away, they never came back. The other thing that was completely different about immunotherapy that some people overlook, if I gave you chemotherapy, if I give you radiotherapy, even if you were a great responder, your tumor went into remission, but we were always worried that it was going to come back, because they always came back. This introduced the concept of immune memory. Just like that polio vaccine you had when you were a little kid, you're protected against polio forever. When your immune system wipes this cancer out, you have immune memory. So if I try to give these mice back this same tumor, it'll never grow there, because the immune system will just wipe it out. So therefore, it wasn't just a transient relapse, or it wasn't just a transient regression that was going to relapse. It was never going to relapse, because the immune system was always keeping it down. So this was a new concept for immunotherapy. Some of the work that I did when I was with Jim just showed that if we block these two main ones, we could really do a lot better. If we block these two together, we could cure 50, 70% of animals that had melanoma. Now, in my career, I've cured many mice of cancer. I'm really good. If you got a mouse, he's got a problem, I can help. But that doesn't always mean the same things, unfortunately, work on patients. So this time, though, we were really glad to see that when we took this to melanoma, and therapy for melanoma has changed so much now, we actually forget, but if we go back to 2010, metastatic melanoma had a median survival of 1 year. About 10% of patients were still alive at 2 years. It was horrible. With this treatment, which is now the standard therapy for melanoma, 2-year survival is 90%. About 60% of patients get below this line of tumor shrinkage. And this group over here, 12 to 15%, are cured forever. All their disease goes away, they're cured forever. And so obviously we want this to be everybody, but this was really effective, and it's sort of driven this huge excitement around immunotherapy. Now again, the problem that I referenced when I was talking is that, look, melanoma, lung cancer, they're up here. When we say hot, it means that they already have your immune system in. Your immune system sees them and it tries to get in there and it does a pretty good job, but eventually the tumor wins by shutting it off. When we reverse that with immunotherapy, really almost half of patients can have really strong benefit. The problem is the majority of patients that walk through the door somewhere like MD Anderson have tumors that have done such a good job locking the immune system out that we call them cold. And that's where we need to get better at treating these cold cancers, because the first drugs that we develop just don't do a very good job. And now I'll give you just one example of how this innovation cycle is working, right? And it's a personal example for me. We did some work at MD Anderson, both in my lab and at a platform that Jason oversees called Orbit, where we developed a new antibody as kind of a second generation of these kind of checkpoints that we talked about. It was licensed to a company, ImmunoGenesis. ImmunoGenesis then, thank you, Texas, got a $15 million CPRIT grant to help develop this as a drug. And now all the way full circle, we'll be treating patients at MD Anderson this November with this totally innovative therapy that isn't going to be available anywhere else until hopefully it works, and then we can share it all around. And I promised I wouldn't show data here, but just to tell you why we're excited about this, this is, again, in one of these tumors where the best immunotherapies we have now just don't work, and this will cure 50% of them. So hopefully, again, this isn't one of those mice don't work like people. Hopefully the people see the same benefit. Why is, some of you investors, what's driving the excitement around, we have literally dozens of new startups in immunotherapy in Houston. Well, these drugs, these PD-1 inhibitors and also CTLA-4, even though we can only treat about 45% of patients, the value of that market in 2025 is estimated to be $50 billion. So given that the majority of people are still here and we don't have a way to treat them, all of the companies that are forming, especially in Houston with the unique mix of research hospitals, patient density, early startup infrastructure, and venture capital, there is this huge financial potential as well that's drawing the money that supports nucleating all these new companies. Now, some companies are focused on what I've talked to you about, which is immune checkpoint blockade, but really because of these unique features and some others that Jason will tell you about, Houston's become one of the world's really top hubs for adoptive cell therapy. And that's his expertise, and I'm going to stop. Thanks.
Great, thanks, Mike. That's perfect transition. So I'm going to tell you a little bit about the other side of immunotherapy, which is adoptive cell therapy. First, a little bit about my pathway that led me to Houston. I'm not only an adoptive Texan. So I started my career studying biology at great academic institutions and was on a pathway in terms of staying in academics, much like Mike has. But towards the end of my graduate career was the genomics boom, and I just felt like there was a tremendous amount of potential to impact patients by going into industry, whether it was biotech or biopharma. And so left academia, and for the next 20 years spent that in small, medium, and large size biotech and biopharma, building organizations to develop therapeutics, therapeutic proteins, monoclonal antibodies that could treat unmet medical needs. This was, then I decided that, well, let me say this. One of the challenges historically over the last 20 years with protein therapeutics was the ability to manufacture them. They were very complex molecules. You couldn't just synthesize them by putting them in a machine and pushing buttons. You had to use living cells to produce these. And so over the last 20 years, we developed advanced manufacturing techniques to control and produce these products. Over the last 5 years especially, although the field has been around for decades, but really over the last 5 years, this new kind of immunotherapy, cell therapy, became more evident. And one of the challenges in that field was manufacturing these products. We knew they had tremendous efficacy, and so that was an area I wanted to get into. And so looked around at various different possibilities, geographically and company-wise, and actually had a lot of opportunities, but chose to come to Houston, chose to come to MD Anderson. And let me tell you a little bit about what we've been working on over the last 2.5 years. So, cell therapy. Mike gave a good introduction to it. This is a picture of Bill, who was one of the first CAR T cell therapy patients. Bill had a type of B-cell lymphoma. And for 10 years, he was on various chemotherapies and different kinds of treatments. And finally, all of those options had run out. This was around in 2010. And so he enrolled as patient number 1 in a very experimental therapy called CAR T, where we basically, this was run actually not in Houston, at Penn, where they took Bill's cells, his T cells that Mike was talking about, engineered them with some fancy techniques and reinfused them. And after 10 years of living with cancer and getting various chemotherapies and getting checked in all the time and just seeing, how his disease had progressed or slightly regressed. 30 days after he had been given his one dose of CAR T, the doctor called him in and said, we can't believe this, but we no longer detect any cancer in your body. And as Mike was saying, this is really the power of immunotherapy. 10 years later, still has cancer-free. And so if we can use the word curative, this is an example of what we can achieve. And that's, in addition to the most important thing, which is Bill is still alive, but it's also the quality of his life. He was given this one treatment 10 years ago. The first week or two that he had that treatment, it was a pretty rough go as your body mounts a huge immune response against this invader. Much like, for us, we've got the COVID vaccine or other things. That first day or two after you get the vaccine, you felt feverish, and that's your immune response. So a massive immune response, but then cancer-free. And so he doesn't have to live with taking a pill every day or going in for chemotherapy and feeling like crap for the next 2 weeks. He's gotten this one treatment, and he's cancer-free, and that's what we're trying to do. Okay. So that's why I wanted to get into this field. MD Anderson has been a leader in this area for decades. And really the two areas that MD Anderson has led in has been on the clinical side, where more cell therapy clinical trials are run at MD Anderson than any other location, as well as on the science side, as Mike described, understanding what works in mice. We call that translating that into people. And then when we give these therapies to people, understanding how and why they respond. And so this deep science knowledge too. And so the clinical and the science were really strong. Now this is sort of shifting to another mode in terms of, for maybe investors or people who are interested in the value proposition as well. This curve that I'll come back to a couple times shows the value of a particular therapeutic on the y-axis over time or milestones of a therapeutic's development on the x-axis. And so, as you can see at the research phase, like Mike said, lots of people cure cancer in mice, unfortunately. And so
it's undervalued.
Yeah, maybe undervalued, but it's very low because there's a lot of people who cure cancer in mice, and it's hard to translate that into people. And so you have to do lots of things to move what you learn. What you've learned works. And that's what's amazing about what Mike has done. What you learn works in mice to translate that into people. As you get through those various milestones, you show in various preclinical models, animal models, that it works. Then, and this is just what Mike's company is doing now, you put together what's called an investigational new drug application, which is basically all the information that says, hey, we've got a great new therapy that we think will help people, and here's how we're sure it's going to be safe, and you show that to FDA. And also how we're going to manufacture that product. Putting all of that together is a huge milestone and inflection point once you get through that, because now you've de-risked the fact that FDA may say, look, we don't believe you, we think this is too risky, or we don't think your manufacturing process is robust enough. And then the next big step is clinical proof of concept, where now we give the product to patients and we see responses. You get through if you get through those steps, there's a very steep value inflection. And typically, the way historically academic institutions have participated in this value curve is at the preclinical stage, and curing cancer in mice or treating various diseases in mice, and then didn't have the resources or the knowledge set to translate that into the clinic. And so oftentimes at academic institutes, that's a point of partnering with pharmaceutical companies or spinning that technology out to a biotech company. what we wanted to do within MD Anderson is build capabilities for cell therapy to internally take these products through this value inflection curve. And so in order to do that, we would complement the traditional strengths at MD Anderson on the clinical and science side with industry scale manufacturing capabilities, as well as a group that understands how to develop therapeutics in a regulated environment in alignment with FDA expectations. And so that's what we set about to do. Thinking that by this organization could take this curve that I showed you before and steepen it. Basically, get to these milestones in shorter amount of time and increase the value of those milestones because we're de-risk things and well set up for future commercialization. And so this was the value proposition that we put together. It was interesting, and it's a good lesson to like keep your eyes open, even though you have a good plan and are executing against that. While our initial idea was to take MD Anderson faculty inventions and turn them into great therapeutics to treat patients, as we were putting together this engine, people asked us, venture capitalists actually asked us, why not do this with early stage biotech too? It's an area that they struggle with. That makes a lot of sense. We talked to some very interesting early stage biotech. Obsidian Therapeutics was one of them. They had also shown that with their technology, they'd cured cancer in mice, but were struggling with how to bring this to patients. The challenging part for companies, for anyone in this space, is that with these treatments, you need the logistics and the choreography of the patients and the manufacturing, because you actually take the patient's cells, transport them to the manufacturing facility, do your engineering and manufacturing there, and then that same batch you transfer back to that same patient. Takes typically between 2 to 6 weeks to do this. Highly personalized, one dose, one patient. And so for early stage companies, very difficult to do this. While we're set up to do this at MD Anderson, we have all the connections now as we built this manufacturing capability along with the clinical capabilities. And so we've partnered with several biotech companies to take this engine and develop innovative cell therapies. The other great thing about this model is, innovation is really hard and elusive. How do you capture that? A lot of it goes on in our academic institutes. A lot of it goes on at TMC, Texas Medical Center. But it can go on in many other places, very much. Obsidian is based in Cambridge, Massachusetts, sort of a hotbed of biotech. And yet now we're able to pull that innovation down to Houston, and we're going to be running clinical trials at MD Anderson with that innovation. And as we develop that therapy, very likely, if it works well, Obsidian would expand in the Houston area. And so it's another way in which we can sort of pull innovation and biotech into this area. So in a relatively short time, this is the CTMF is the group that I told you about. We've developed a number of capabilities in manufacturing and then applied those to both faculty projects, MD Anderson faculty projects, and biotech innovation. And so in the next year, we're going to take 5 novel cell therapies and bring those to patients, which is pretty amazing to do for any company, and I think will give opportunities. The most important thing is that it'll give opportunities for more patients like Bill to have curative potential, but as well for the local biotech seen in Texas and Houston specifically, will offer many shots on goal, many opportunities for success. So that's why I wanted to share with you a little bit about our story. Thanks for listening.
All right. Thank you, Jason and Mike. You know, it's interesting. Just there's one thing that I would layer on top of one of Jason's slides with the value curve. There's also a risk curve that kind of goes in the opposite direction. It's almost kind of like supply and demand. And you know, it's interesting. There's coming out of Anderson a few years back. There was a CAR-NK. It was a cord blood-derived natural killer cell engineered program. And we tried to find that a home for years. And it was interesting, once it got into the clinic and it was past all of the manufacturing hurdles and FDA, you know, IND files and all that sort of thing, they were very fortunate in the first, I think, 3 or 4 patients that were dosed to see a clinical response, and not even, oh, that looks pretty good. I mean, you see the before and after photos and you don't even recognize the patient. Full body CT scans, loaded completely with tumors and completely clean. I mean, it's just jaw-dropping. And that program went from, it's too risky, it's too different, it's not CAR-T, to literally a feeding frenzy with potential licensees and investors that were, you know, essentially kind of beating the door down. So I just can't tell you how exciting it is to have not only the capabilities, but the folks that have sort of seen this in the past of what success can look like, and you know, to kind of keep building on that model. One question I was having, I was just jotting some notes and stuff down in addition to some other thoughts I had earlier. People talk about CAR-T, CAR-NK, CAR-M now, I was reading recent articles on that, TCR therapies, endogenous Tcell, TILs, cancer vaccines. I mean, I realize they're all variations on a theme, but I mean, if you had to kind of rank order those, I realize it's almost like how high is high, but what do you see really truly moving the needle? And I realize that you have to handicap these because they've all been developed at different time frames.
I'll touch on that. I'll leave part for Jason too. You know, the tricky part is when you say moving the needle, and Jason alluded to this, the difficulty in manufacturing the T-cell therapy. So the thing with T-cell therapy is, I'm going to take your T-cells, I'm going to re-engineer them, I'm going to grow them up to trillions. You know, if your body's naturally trying to fight up the cancer, you might develop tens of thousands or hundreds of thousands of cancer-specific T-cells. Sometimes it's not enough. I'm going to fix that problem by taking them out of you, growing up a trillion, literally a trillion, and giving them back to you. That should be enough, right? That's the thinking. The problem is that's a very expensive personalized therapy, right? And so what moves the needle is a balance of how hard and how expensive it is to scale that up if you're thinking about an FDA-approved therapy that everybody's going to be able to get everywhere, but you're going to have to reimburse it for $1 million to your insurance, versus balancing things that may be ways to achieve the same end but with less costly manufacturing. And so with all that being said, look, the NK cell therapies have started to look really good, and people have been able to develop either stem cell based or allogeneic, meaning they can come from any donor. So the trendy term is off the shelf, right? So a lot of these personalized therapies, if you can give them from an off-the-shelf source, I don't have to take them from you, the patient, make you wait while you're sweating bullets for 4 to 6 weeks while I prepare this for you, but you can't get any other therapy, and then give them back to you. That's super expensive, but amazingly effective, right? If I can find a source for that is from a general off-the-shelf donor, then I've got something now that packages the efficacy of this personalized therapy, but along with a scalability that maybe doesn't exist here, despite the improvements that have definitely been made. So I think some of the NK-based CAR therapies look really good. The T-cell therapies are to some extent catching up, because if for a CAR T-cell therapy, I can give it to anybody. So if it's for CD19 positive B-cell lymphoma, I can give it to anybody that's got CD19 positive lymphoma of any kind, right? If it's a T-cell therapy, your T-cells, you have to have the right HLA, right? It's like we're matching organ donations, right? So they're a little bit more narrower, and so they're catching up. But it's harder to scale them up to large amounts of people, although there's some that have been very effective. And then cancer vaccines don't really, they fit in a different place. They're not for treating advanced cancer patients, really. I think people have learned that lesson. but they can be remarkably, they have almost no side effects, right? CAR T cells can kill you if we don't manage the side effects, and it's very aggressive management. Cancer vaccine's not going to hurt you, and so, but they are very effective at mobilizing the responses. So how about when I'm going to do surgery to take out your cancer, but there's a big chance it might relapse, I'm going to give you a cancer vaccine. You might be cured by the surgery, so I don't want to give you something that can hurt you or kill you. I want to give you something that's sort of side effect neutral, but can help you, be progression-free, be recurrence-free longer. That's where cancer vaccines can fit in really nicely. So it's a little apples to oranges, but I like the off-the-shelf NK CAR, allogeneic T-cell CAR. I think those are at the top of that bucket. But Jason may think differently.
Well, no, I think that was a great overview. And I think there's, first I would say, just like both these guys pointed out, the bottom line take-home message is it's a super exciting time because there's so many opportunities, so many different modalities, so many different options that are all being developed. So I think that's the main take-home. Like, it's going to be a little hard to tell who's going to be the winner, but at the end of the day, the patient's going to be a winner because we're exploring so many different modalities and so many of them have shown efficacy. There is this, I see it as this like two extremes, right? One, you can have therapies that work that are difficult to manufacture. On the other side, you can have things that are straightforward to manufacture, but still have to be proven that work. Like I said, industry is developing all of the above. As someone who's worked on manufacturing in complex biologics for 20 years, I like the idea of, give me something that works, and then I'll pound away at figuring out how to make that more cost-effective, a more robust manufacturing, figuring out what are the right cells. One of the challenges we have is, as Mike said, trillions of cells, some of these therapies. It's trillions because a lot of those cells are bystanders. They're sitting around watching their friends do all the work. So if we can figure out how to make them more potent and give lower doses, give 1 million cells instead of 1 trillion cells, but make sure it's the right 1 million cells that are really potent and really fired up. That'll bring down the cost of goods tremendously. So, it's much easier to spend a lot of money improving manufacturing when you've got something that's curing cancer than to spend a lot of money to develop something that's easy to manufacture but may or may not work. At least that's the way I sort of look at it. That's very simplistic, but that's how I think about it.
Okay. No, I'm glad both of you brought up the expensive things. That was a question that I had just as far as, not necessarily elephant in the room, but I mean, I don't think the system can absorb too many, $400,000 treatments, and then with all the supportive care and everything on top of that. But
you know what's interesting, Larry, because that comes up all the time in cell therapy, right? We all know, right? $400,000 treatment. Oh my God. On the other hand, how expensive is it to give someone chemotherapy for 6 weeks and have them come in for 6 months every 6 weeks and have them come into the hospital? How much does it cost society for them to be sick for 2 weeks after every dose and that they call in to work sick for all that time? And so while these are tremendously expensive at this point, balancing that with a one and done and potentially cured can offset. There's a lot of value that can be achieved through that you don't pick up necessarily right at day one, but over a period of months and years of curing something that the cost of, I would be interested to see for Bill what the cost for the first 10 years of his cancer treatment were. Probably pretty expensive.
10, yeah, no, that's an interesting way to put it. Yeah.
No, I mean, it's true. That's often overlooked. I think one other perspective to keep in mind, and this is just sort of maybe the philosophical, but from the engineering and manufacturing standpoint, the ones that rise to the top for me, sort of looking into the future, we've learned a lot. We know a lot about the reasons T cells don't work when they get in the tumor microenvironment. And individually, we've learned to engineer them around those problems, right? Recognition, we can engineer things like CAR so they recognize the tumor better despite the tumor trying to hide. But again, I mentioned they sort of poison their surroundings. It's depleted of oxygen. T cells need oxygen. We've learned ways to metabolically engineer T cells so they can actually survive in those environments. There's certain things that suppress them. We can engineer them so they become insensitive to those things. The real key when we have an off-the-shelf therapy that we can treat everybody with a high enough success rate that nobody really cares what it costs, and then we can just work on pressing the costs down. For me, I really like some of the technologies that simplify the multiple engineering, right? A lot of what we have now, I can afford to make one change. I can put that receptor that allows it to enter, but I can't engineer it to survive better once it gets there. Or I'm using Tcell, which is just taking your tumor-infiltrating T cells that have failed out, growing them up, and so I can do one thing there, and they already recognize, so maybe I can put something in there so they're harder to shut off, but I can't do it all in one setting, right? I only get one change. There are some platforms, for instance, now that are stem cell based, where because it's a stem cell and it's a stable pool that will generate as many of these cells as you need, it's easier to make multiple changes, right? Now I can sort of do it iteratively. I have my third generation sort of cell line that's stem cell based, where I've made all of these changes. I really see value there sort of in the 5 to 10 year future, because we do have knowledge of how to engineer the ability to make that million cells, not trillion, that million that are really going to do all the work. But we need an engineering platform that allows us to make enough of those changes safely and at a reasonable enough cost to manufacture. I think we will get there.
Yeah. The buzzword nowadays is like synthetic biology, and that is like touching on what Mike said, where you basically, where you can do 4, 5, 6, 7, 10 different changes to a cell, give it all sorts of different properties to make it a fair fight with the tumor that's doing all sorts of things.
All right. Thank you. A couple of questions. You've kind of touched on this a little bit, both with the, I guess, the pillars of treatment. I don't know what that slide is called, but not in the big scheme of things. Not too long ago, bloodletting and what is it, trepanation, and other these horrific medical procedures were standard of care.
The old imbalanced bodily humors.
Yeah, those and the vapors. Yeah. So looking down the road, do you think kind of traditional chemo is going to have that same sort of, not stigma, but like, oh, gee, I can't believe we used to poison patients, and practically kill them that way. But, so looking, I don't know, 10, 20 years down the road, how do you see that, I guess, treatment option modality versus kind of what the doctors have now? How do you see that changing?
Yeah, I'll go for it. I mean, Larry and I often use the leech analogy, and without disparaging anything, this is the best we could do. Chemotherapy was the best science we had at the time. But if you look forward 20 years, and that's part of the reason why I wanted to get into cell therapy, to accelerate the advent of not only individual therapies, but this entire field. Because this is, in my mind, it's the future. If the only thing holding us back is manufacturing costs and processes and things, we're going to get there. Antibodies 25 years ago were awful to produce, and we spent billions of dollars making that more amenable. It's going to happen with cell therapy, and it's going to be the case that I think that is the future. And 20 years, I don't know whether that's 20 years, 30 years, whenever, we reach Star Trek type proportions of things happening like that. But then we will look back, and say, wow, remember when they used to give poisons and that they slightly killed tumors more than they killed, stomach epithelial or hair cells or things. And now you're giving very personalized medicines that are so precise and so targeted that they are curative. It's going to happen.
Yeah, and all, in the intermediate time.
Yeah.
right? In the
That's the
intermediate
thing.
time, one thing that has happened by adding that pillar of immunotherapy is it's made those other more archaic pillars a little more useful in some ways. So the midterm future for chemotherapy, is not that we're, retiring it tomorrow. It's that we've learned there are certain chemotherapies that are actually less harmful to your immune system and are really good at actually killing tumors in a way that kind of inflames your immune system to recognize and attack them, even can work, sort of hand in hand with your immune system. And that maybe we don't have to give terrible, side effect inducing doses of chemotherapy to induce the effects that are really beneficial from that standpoint. I mean, one of the revelations that came actually, I'm an immunologist, so I'll tell you this. If you use a lot of the chemotherapies we use in mice that don't have an immune system, they don't work anymore. So what we learned is that actually even chemotherapy, a lot of the most common ones, the ones we use for breast cancer, the ones we use for prostate cancer, they actually rely on your immune system to do a lot of the heavy lifting after they're doing this initial kill of the tumor, if you actually want to see a real benefit. So what we're learning to do is tune those a little better. So they're not so bad for you. We don't need them to do all the heavy lifting. We pair them with an immunotherapy. They can sort of jog that process, and the immunotherapy can provide that longer term effect that wasn't there before. Now that's kind of the midterm. I agree, I was, in my distant past, I have a little bit of a computer science background too, and I dream of kind of a Moore's Law type time for cancer therapy, and I think we're almost there, where maybe we can double our response rate every year. I think it's coming, and so, after 20 years of that, are we going to be giving people chemotherapy in almost any setting? Probably not. I think it is radiotherapy, not so quickly out the door, because we've learned to make it much more targeted in a way that actually can work very well with a lot of these other things. Targeted therapy also, we're learning how to better reverse really the very key changes that have made cancer cells into a cancer, and I think there's a place for that in the future as well. But yeah, chemotherapy is probably going on the shelf. We are going to talk about,
Okay.
putting our poisons away.
And this is something that touched on that I just love about biology and science in general, but especially biology is, when Mike showed that slide about the 3 pillars and then immunotherapy added as the 4th, it wasn't like people, if you asked people 30 years ago, 30 years, what about immunotherapy for cancer? Actually, 95% of people, maybe more, would have said, no, the immune system is not involved in tumor surveillance. Mike and Jim Allison can tell the stories of, at the big cancer conferences, there were plenary sessions all about the latest, greatest new chemotherapy and how this is going to revolutionize treatment and extend patient's life by a month.
The immunotherapy session was in the basement between the bathrooms.
Yeah. So there's this little cadre.
Sadly, that's not a joke.
There's this cadre of people, amongst them, who would huddle together in the basement between the bathrooms, talking amongst themselves.
We could drink as much coffee as we wanted. It was fine. Lots of access, but still.
It's right there.
And so if you asked that question 30 years ago, you got a 100% different answer. That immunotherapy is not involved. Immune system isn't involved in tumor. It's too much like self, and the immune system just doesn't see it. And you flash forward not that long, 10 years after that, and it's this amazing new potential. And that ability to I think for everyone, don't assume we know everything and it's just a matter of time. We have to challenge all these assumptions constantly and turn things on their ear. And that's what I just in general love about biology and that we're still scratching the surface of how this body works.
Okay. I had not, I'm sure, certainly familiar with Moore's Law. I had not heard the Mike Law before, but maybe that should be.
I think you definitely think of something better to call it.
Copyrighted immediately. So we're gonna have some time for some questions. We got about 10 minutes left. So if there are microphones both on the side and then one at the back as well, if there are any questions. While people are kind of queuing up, Mike, just real quickly, you've had the benefit and the misfortune of being in the academic environment and helping spin a company out of a place like MD Anderson. Just quickly, any words of wisdom for any budding entrepreneurs in the audience? Don't do it.
Wisdom may be pushing it, but I can share my scars.
No,
I mean, I think it's funny because you're on the academic side, you have a view of things, right? And one of those views was, look, I showed, back in 1996, no one else believed us, Jim had this data saying, look, all you got to do is block this CTLA-4 and tumors go away. We should really do that in people. Well, that drug got FDA approved 14 years later, right? I mean, it took that long for we're all screaming our heads off, and it took that long for it to happen. And so, when I got into this, I had the academic view, which is, I don't know what these people have been doing, but I can do this faster. I can do this in 2 years. We're going to be treating patients. This looks great in mice. I'm going to find out if it works in people, and I don't know why these people take so long. Well, I've learned that there are reasons, and there are, you go to the other side and you get a perspective. Well, if you actually want it to work, if you actually want to get through all the regulatory hurdles, if you actually, want to come out of a phase 1 trial with data that allows you to move forward and get it closer to where you can give it to large numbers of people and get registration, there are certain things that you have to do, and there are certain timelines that you can't accelerate. Now, still, I think we cut it down to about 4 years, so it's not 14, but it did, as much as I thought I was smarter, it takes a long time, and you sort of learn both perspectives. But what I could say is, look, if you have a good idea, don't be afraid to talk to people about it. That people will be happy to, give you feedback. Talk to people who have started small companies. Does this look like the kind of thing? What am I missing that I don't have? And there are things you may not think about in terms of, where the IP is or where you're going to get your initial money just to start the company. I knew some things. There are certain things I didn't know. I had to call my old mentor from graduate school, and because I was like, do I own this company that I started, or do I have to put a bunch of money in, because I don't have very much money. I think this is going to work. I want to buy some of this company. He's like, you started the company. You own 100% until you start giving it away. I'm like, oh, that's cool. Neat. I can put my checkbook away. I thought I might be able to afford to buy 5, but now I've got 100. So anyway, I mean, there's a lot to learn, but I've found that people, both in the venture capital space, in the sort of entrepreneurship space, it's definitely worth it to talk to people and get feedback about, and don't be afraid to put it out there, because, I got a little push, and I'm so glad that somebody gave me that little push to get it started, because, I don't know what's going to happen, but we've got 3 drugs we're going to test in patients in the next year, and for me, that was my goal, right? I wanted to see these things actually get to people. So.
And I'll just get to the question, but just to echo and accentuate what Mike said, you got to push. These things are just not going to happen by themselves. They're not like a moment. You have to generate that momentum as an entrepreneur yourself. Even the CTLA-4 wouldn't have, as Mike was saying, that these guys were screaming their heads off. If they weren't screaming their heads off, probably wouldn't have happened, or would have happened much, much later. So the empowering thing is you can make a difference, but it won't just happen. You'll have to put yourself into it and really push.
All right, thank you. All right, we've got a couple of questions. So sure, if you want to step up to the mic, please, and introduce yourself real quick and then ask your question.
Excellent. My name is Matt. I just had a question about how to accelerate these things, because as you alluded, it takes a long time once you have the mouse data to get it into the patients and prove it out in an FDA-approved kind of a way. And really that trial process, especially for cancer, is some of the longest and most expensive trials that we've got. At the same time, chemotherapy is radically different from the immunotherapy stuff that you're putting there. So my question is about the endpoints. How do you tell the story of that difference and the difference of the cancer journey and all those things through the traditional lenses of objective response rate, total mortality, those types of things? Are you reframing that? Are you pushing it all to progression-free survival? Are you trying to do some different things? And then also, how are you trying to put in quality of life measures into those trials?
Yeah, those are all super good questions. The marginally unsatisfying response is it depends on what stage of patient you're going to treat, what your metrics become. The traditional view that you would be taught is phase 1 trials are just for safety, right? We want to find out if it's safe. Unfortunately, if you talk to anybody who you hope to give you money, if they're actually going to give you money, they want to see whether it's working or not during that phase 1 trial. And so you're always trying to balance those things. For the example I gave with ImmunoGenesis, where we have a drug that we think is sort of better than the existing PD-1 drugs, we know it's at least as good. So the challenge for us is to show that we're differentiated better. So we have this advantage of actually, we're in a known area. We know it's going to have some efficacy. So the trick is really, and this is a trick across all of those spaces, find the right patients to show that you're different or that you're working in the patients you think you're going to. For us, that involves some biomarker screening up front, but this is really important across all. I see a lot of good drugs fail, even at phase 3 IDO inhibitors, because they treat the wrong patients. They don't narrow their patients to the ones that the drug is going to be right for by screening them up front because it's too expensive or too laborious, et cetera. So yes, you can use objective response as a metric, but make sure you have the right patients. Or, there are some new therapies where, like some of the Tcell therapies where they really have to do safety first, but safety is usually small and then you're expanding into efficacy. What we're doing a lot of too these days is neoadjuvant. Neoadjuvant means we're treating patients that can, with a non-toxic therapy before surgery, things like that. Those therapies can be phenomenally effective because we finally get to treat earlier stage patients. The metric for those patients is, look, they're going to get surgery. We know 50% of them are going to relapse within 6 months. So there, you're just following and saying, did less of them relapse? And when I took out that chunk of tissue at surgery, did it look like a lot of the bad cells were gone? So some of those metrics really, float with where you're trying to treat patients.
All right, thank you, and appreciate everybody, your attendance and attention this afternoon. Thank you.
Thanks, everyone.